Right here we have the cell cycle. First we start with G1, which is the growing phase, and then we get into the S phase where it stops the growth, and then it copies the DNA, and then we get into the G2 phase where it continues to grow, and then we get into mitosis.
Alright. In biology, mitosis is used to describe the division of one parent cell into two daughter cells. Mitosis is imperative in the repairing and growth of bodies and making sure that all the cells receive the same DNA from the first cell. In mitosis, it is important to know that there are haploid and diploid cells. In eukaryotic haploid cells, it basically means that it has one copy of all of its DNA. In eukaryotic diploid cells, it means that it has two copies of its DNA. It is easy to remember by remembering that di means two. Mitosis is conducted using the IPMAT process, which is part of the cell cycle. The end goal of mitosis is two cells that are genetically identical, which can be generally helpful, but mitosis can also sometimes end up in tumors, which form cancers. And that's it.
Unwinds the DNA into two individual strands. SSBs coat the DNA strands to prevent reannealing. Primase is an RNA polymerase that synthesizes the short RNA primers needed to start the replication process. Polymerase strings nucleotides together to form a DNA strand. The sliding clamp helps hold the DNA polymerase onto the strand, and RNase H removes the RNA primers, and RNA ligase links the short strands together.
First, helicase comes through and unzips the DNA strand to two separate DNA strands. Next, SSB coats both the DNA strands to prevent reanointment. Then, primase comes through and lays short RNA primers so that the polymerase can work off of them. The sliding clamp holds the polymerase onto the DNA strand while the polymerase goes through and replicates the DNA. Next, RNase H comes through and removes the short RNA primers. And then polymerase continues. And finally, ligase comes in and joins the two DNA segments. Now, we have two DNA strands. The cell cycle begins with G1, S, and G2, which are known as interphase, and then goes into mitosis. Mitosis consists of 5 steps: prophase, metaphase, anaphase, telophase, and cytokinesis. Prophase is where the loose chromatin within the nucleus begins to condense into chromosomes, and the centrosomes split and go to opposite sides of the cell. In metaphase, the chromosomes go to the middle of the cell and the spindle attaches onto them. In anaphase, the spindle pulls the chromosomes apart to opposite sides of the cell. In telophase, a new nuclear envelope develops around each pair of the chromosomes, and the chromosomes begin to unravel into chromatin. Finally, in cytokinesis, the cell divides in two.